couple of things. Here we're going from the least packing efficiency to the most. And for equal sized spheres, this is the highest packing efficiency you can get, face center cubic. With face center cubic, you're going to have the least amount of empty space, the most amount of occupied volume. And so where do the atoms touch? Do the atoms touch along the corners? No. The atoms touch along what we call a face diagonal. This is a face diagonal. This was a body diagonal here. And so along the face diagonal here, there are 4 radii when we slice and dice the atoms. The face atoms are going to be cut in half for the cube. And so half of the face atom resides in this cube, the other half resides in the cube above. One thing you want to note is something called the coordination number. If we go to body center, the coordination number is fairly straightforward. This red atom here is coordinated to how many white atoms? 4 above and 4 below, so a total of 8. In fact, the corner atoms, if you extend the cubes out, are also 8 coordinate. So all the atoms are 8 coordinate. What's the hybridization for 8 coordinate? You need 8 bonds. sp3. Not enough. sp3d2 will get you 6. sp3d4, right? But sp3d4, that's for covalent bonding. Usually what happens in metallic bonding is non-directional. You know, with metallic bonds, you don't have a tetrahedral arrangement of sp3 covalent bonds, or a trigonal planar arrangement of sp2 sigma bonds. With metallic bonds, it's thought to be, you know, the electron C model would be the cations here surrounded by a sea of electrons. So the bonding is non-directional. And so metals, one of the properties of metals is that they're malleable. That is, you can distort them easily because all you're doing is you're pushing the atom at a different angle. Well, if you push this from, let's say, this angle to this angle, is it going to break the bond? No. Whereas if you did that in diamond, if you push one carbon atom this way, will that break the sp3-sp3 overlap? Pushing one carbon atom over this way? Yes. But in a metal, since the bonding is non-directional, no problem. You could do that. It's not going to be maybe the most stable, but you can do it. What's the coordination number in face center cubic? So count it. How many atoms does it touch? Let's pick this red one. 1, 2, 3, 4, 5, 6, 7, 8. Then you have to project out 1. 10, 11, 12. 10, 11, 12. It turns out that the coordination number is 12. It's hard to see, but it's 12. One way you could see the coordination number better is to tilt this like this. This is what you just built. Take the body diagonal and tilt it so that the cube stands on a corner like this. Can you do that? So just grab it by the base and leave the base, but just tilt it so that one corner atom goes to the bottom and the other corner atom goes straight to the top. Do you see it now? The other corner atom being along the body diagonal. Okay, now what you're going to look for are layers. Do you see the layers? The one corner atom is layer 1. Then these atoms, the blue ones, we'll call layer 2. The red ones we'll call layer 3. And finally, the other corner atom is layer 1 repeated. Do you see that? And so this is what we call the ABC stacking pattern. The ABC stacking pattern is one of the most efficient ways of stacking this. If we have cannonballs like this, layer A, B, C, A, B, C, A. And we pack the cannonballs so that they're closest packed. This is what a closest packed layer looks like. Take a look at the yellow layer. A closest packed layer would have one atom coordinated with 1, 2, 3, 4, 5, 6 atoms in the plane. And so it's coordination number 6, yellow. But look underneath this one. What do you see? I see 3 reds. 1, 2, 3. Do you see it? And then the next layer is going to have 3 more. The 3 more could overlap with the bottom layer. That is, they can be in sync. What do you call it? Superimposed. Or, if you put them in these 3 gaps here. Or you could put the 3 in these empty ones where it doesn't superimpose. If you put it in these empty ones, we have the ABC stacking pattern. Layer A, which is closest packed, layer B, layer C. That's shown here, A, B, C, A. That's shown here, A, B, C. Take a look at these bigger layers. Do you see how close packed the atoms are? And then we're back to the other corner, and it just repeats. So again, this is called cubic closest packing. Cubic closest packing has the highest packing efficiency. Do you know what the highest packing efficiency is in terms of percentage? This would be it. Highest packing efficiency in terms of percentage would be about 74 or so, in the mid 70% range of occupied space. What about the unoccupied space? The unoccupied space is very important in face-centered cubic. And so this is what we're going to do. We're going to take a look at what we call the holes. We have 2 important holes. The trigonal hole is not important. The tetrahedral hole and the octahedral hole are the most important. You can see a tetrahedral hole. How do you see a tetrahedral hole? Put the cube back down and then pull a corner atom off. Pull a corner atom off and then stick a tiny pink atom in that little gap formed by 3. It doesn't go on a rod. You put it, gently put it into the gap formed by the 3 and then replace the corner atom. Do you see now you have a pink ball stuck in the, what we call the tetrahedral hole. Did everybody accomplish that? You got to tilt it. So pull up the corner atom, tilt it so that you get 3 atoms laying flat horizontal. And then put a pink one. Put a pink one in this gap right here. Formed by the 3. And then cover the corner. And then cover the corner. Did everybody, were they successful at trapping a pink one in the underneath the corner atom? Now how many corner atoms do you have? 8. Therefore you have 8 tetrahedral holes. So you could actually put 8 pink ones, but we aren't. What's the next type of gap? The next type of gap is in the dead center of this. Is there anything in the middle of this cube? No, the middle of the cube is empty. In the middle of the cube, that's called a octahedral hole, right? because it's surrounded by 6 atoms. We have an octahedral hole in the center, and you see in the center of each edge, in the center of each edge is also an octahedral hole. So take a look at this crystal here. This crystal is sodium chloride. Sodium chloride is the face-centered cubic lattice of chlorides, with the sodiums occupying all the octahedral holes, including the one dead center, right there. And so you got the face-centered green lattice with the sodiums in all the octahedral. Octahedral because it's coordinated 6. Coordination number would be 6 for the cation. The coordination number in a general face-centered cubic is 12, the highest coordination number. What? Okay, that's ionic. But what crystallizes in face-centered cubic here? What crystallizes in face-centered cubic would be like gold. Gold is very malleable. And so gold has a coordination number 12 bonds. It's not very directional. And so you can distort these a little bit without breaking the bond. Okay, there's, I said face-centered cubic is the densest. That's an A, B, C, A stacking pattern, like this. But face-centered cubic is also called cubic closest packing, CCP, cubic closest packing. But we also have the alternate pattern, ABA, which is equally dense with the exact same packing efficiency. It just has the ABA pattern. Well, the ABA pattern doesn't fit in the cube as the repeating block. It's hexagonal closest packing. A hexagonal unit cell looks a little different than a cubic unit cell. Oops. Where's the hexagonal unit cell? Here. This would be the hexagonal unit cell here. This is kind of this trapezoidal thing. Here. There's more to it than is showing. But that's hexagonal closest packing. ABAB. Cubic is ABC. How are these crystal structures determined? X-ray diffraction. X-ray diffraction, you can determine the structure, you know, how the atoms, ions, etc. are arranged in the solid state. X-ray diffraction takes advantage of this constructive and destructive interference of waves. As the waves are reflected off these surfaces of atoms here, these are the atoms, planes of atoms, it depends on the angle. And so if the angle's a little bit off, then the waves are out of sync, out of phase. They cancel each other out. And so the waves are only in phase at certain angles. Those angles are called the Bragg angle. We have Bragg's equation, but you don't have to worry about that. Here again showing the tetrahedral hole under the corner and one of the octahedral holes in the center. All right, and then there are others. For example, zinc sulfide, zinc ions in the tetrahedral. Not all of them, half of them. Calcium fluoride, fluoride ions in all the tetrahedral. This is a different unit cell. Here, not cubic. All right, so some properties of these solids are going to depend on the strength, you know, of the forces holding the solid together. And so we have metallic solids with metallic bonds. Metallic bonds are not localized. Metallic bonds are delocalized, and therefore they're not very directional. Metals vary in... Did I already talk about this? No? No, okay. Metals vary from extremely soft to extremely hard. Like sodium you can cut with a butter knife. You know, tungsten you can't. Tungsten's hard, sodium's soft. The melting points vary. Sodium melts at a very low temperature. Tungsten melts at a very high temperature. Metals are lustrous, ductile, malleable, good conductors of electricity and heat, because, especially electricity and heat, because of delocalized electrons. Ionic solids have electrostatic attractions between ions called the ionic bond. They're hard, moderate to high melting points. Non-conductors as solids, but good electrical conductors as liquids. Many are soluble in polar solvents, such as water. Network covalent would be like diamond, graphite, silicon carbide, aluminum nitride, silicon dioxide, very hard. Hard to melt. Non-conductors, except for graphite, of electricity. Molecular solids are always soft, like ice. Slow to moderate melting points. Some examples of molecular solids. Some of them have kind of high melting points, like asphalt, wax. Polar solids, you got dispersion. That would be frozen acetone, frozen chloroform, frozen HCl. Those aren't going to be very hard. Hydrogen bonded, we already know one quite well, water, properties of water. All right. I talked about the lattice energy. Lattice energy and bond energy aren't exactly the same, right? And so how do you figure out what the lattice energy is? Well, the way you figure it out is using Hess's law. All these other values here are known. And so we could use Hess's law and add up all these known values to figure out what the lattice energy is. And so the lattice energies, that is these bond energies, cumulative bond energies, can be calculated here using Hess's law. So that's the end of chapter 12 there. All right, so what we're going to do is, since I don't have enough time to show, actually I probably do have enough time to show these, but I'm only going to show one. What are you more interested in? I was supposed to show both these videos, but maybe I'll show one in Chem 1B. Molecular structure and health, or crystals and their structures? Crystals using X-ray diffraction. Molecular structure, looking at what we've been talking about, the shapes of molecules. Crystal structure. Molecular. Let's take a vote. Molecular. Crystals. 3-2. Sorry, crystal structures. You guys will have to wait. So we're going to check out a video about some application in chemistry right now, or some of the concepts we've been studying. These are concepts from the very beginning. So yeah, go ahead and put everything back to the way it was.
This is a production of the Chemical Education Material Study. The number and variety of living systems is remarkable. More than 1 billion species currently exist. We now know that a principal factor in this large number is the billions of ways that 4 rather simple chemicals can vary their sequence in forming DNA genes. We are also discovering that many illnesses can be identified at the molecular level. This provides information for the design and synthesis of molecules, which can be used to treat the disease. If we examine living cells under the microscope, like these plant cells, or these bacteria, or this animal tissue, we can see the movement, growth, and cell reproduction, which are the result of successions of chemical reactions. In attempting to control these reactions, physicians have used various substances, some from nature and some synthesized in the laboratory. Many of the compounds found to be useful were discovered by trial and error. One such compound was sulfanilamide. Let's demonstrate the effect of sulfanilamide on bacterial growth. A nutrient medium in a petri dish is used to sustain growth of bacterial organisms. These bacteria, harmful to humans, are carefully transferred from a stock preparation. They are gathered in a wire loop and streaked onto the nutrient medium in both compartments of this dish. A small piece of sterilized filter paper is wet with a sulfanilamide solution and placed on the bacteria in one compartment of the petri dish. The dish is placed in an incubator for 24 hours. During that period, seen here in time-lapse photography, the bacteria multiply and become visible as large colonies everywhere except in the area around the paper disc. The clear zone shows that bacterial growth has been stopped. This inhibition is due to the sulfanilamide from the disc. How does sulfanilamide stop the growth of these harmful bacteria? First, let's examine the function of one of the key molecules that exists in the bacterial cells. It is called para-aminobenzoic acid. Using a geometric symbol retaining the general contours of the molecule, we'll follow its action within the cell. In the cell with the acid is an essential bacterial enzyme. In order for the enzyme to perform its active role, it must first combine with the para-aminobenzoic acid. Only then can the enzyme catalyze the further reactions necessary to cell life. When we examine the structure of the acid molecule and compare it to a molecule of the drug sulfanilamide, we find that they are much alike. This large portion of each is identical. The electron charge distribution in each molecule is also similar, and their general configurations are the same. Using geometrical representations again, let's include sulfanilamide in the bacterial cell along with the normally present acid molecule. The sulfanilamide too can react with the enzyme. But the sulfanilamide not only fails to activate the enzyme as a catalyst, it also prevents the acid from doing so. Thus, metabolism stops, and the bacterial cell dies. The effect of sulfanilamide on cell growth was found by trial and error. But the discovery that sulfanilamide is structurally similar to an essential molecule accounted for the action of the drug. This principle of structural similarity helps us to predict which drugs might be effective against specific diseases. Consider, for example, the use of drugs in the treatment of cancer. By means of tissue culture technique, human cells are kept alive for microscopic observation. Here are normal human skin cells under the microscope. In time-lapse photography, cellular activity appears accelerated. We know of very few differences between the chemistry of normal cells and these cancer cells. One remarkable difference, however, is that the cancer cells grow faster. And another difference is that during the faster growth, The cancer cells use abnormally large amounts of a substance known as uracil. Can a structural modification of the uracil molecule be used to disrupt the fast growth of the cancer tissue? Let's follow some of the steps a biochemist takes in answering such a question. He is familiar with the visible parts of a single cell, like this one undergoing division. We see a set of dark ribbons in the middle of the cell. These dark objects are chromosomes, and they are intimately connected with the process of cell growth. The biochemist has learned to collect chromosomal material from cells and has found that it is composed of deoxyribonucleic acid, or DNA. Because DNA is a large, complex molecule, it is helpful to break it into its component parts and identify them. These component parts can be separated from each other by the technique of paper chromatography. After treatment with an acid, a small drop of the chromosomal material is placed on this large piece of filter paper. The paper is rolled into a convenient shape for the next procedure. We have selected a solution in which the chromosomal material is readily soluble. This is placed on the bottom of a glass cylinder. The solvent is drawn up the filter paper by capillary action. After 12 hours, the solvent front has risen this far. The filter paper is removed and allowed to dry. It is then rolled in the opposite direction and put back in a different solvent. This will ensure a further and better separation of the molecular components. These components are not visible under ordinary light, but if we place an ultraviolet light under the dried paper, we can see that the solvent has distributed the components of the chromosomal material. This distribution has occurred because the solvent front competes with adhesion to the paper for the chromosomal materials, so the components move according to their molecular size and solubility. Thus, different compounds derived from the chromosomes are separated. We'll cut out one of these marked areas and dissolve the material from the paper with another solvent. Now we can analyze the dissolved material with an infrared spectrometer. A characteristic spectrum is produced by each component separated in the chromatography procedure. The absorption spectrum produced by one of these extracts is compared with the known absorption spectrum of the chemical thymine. Apparently, chromosomes contain thymine. Similar analysis of the other components shows that each has its own characteristic spectrum. Identification shows that besides thymine, chromosomes contain adenine, guanine, and cytosine. How do our uracil molecules, which are present in such large amounts in cancer cells, relate to the constituents we have extracted from the chromosomes? We know that these constituents, along with other building blocks, are necessary to make DNA molecules, which in turn have a part in forming the chromosomes. Biochemists have shown that uracil is the substance from which thymine is formed. Without uracil, thymine will not form, preventing the synthesis of DNA, which in turn prevents the formation of the chromosome. When we examine the molecular structures of uracil and thymine, we find that there is but one position in which these structures differ. To convert uracil into thymine, a hydrogen atom here must be replaced by a methyl group such as this. Let's symbolize uracil with its hydrogen atom like this to follow its action within a cell. First, a chemical group called a sugar phosphate residue becomes attached to the uracil molecule. The modified uracil compound then forms a complex with an enzyme. Somehow the enzyme converts the hydrogen into a methyl group to form the thymine compound. This molecule is then freed for eventual synthesis into the DNA chain. How can we disrupt this mechanism and stop cell growth? Clearly, one point of attack is the original uracil molecule. This molecule can be altered in many ways. One of the most promising has been to replace this hydrogen with a fluorine to give 5-fluorouracil. 5-fluorouracil has a number of advantages. First, the hydrogen and fluorine are almost the same size, so that both molecules should fit into the enzyme. Second, the chemistry of a carbon-fluorine bond differs from that of a carbon-hydrogen bond, so that the enzyme might find it difficult to replace the fluorine with a methyl group. And finally, the changed polarity may allow the fluorine compound to adhere more firmly to the enzyme than would a uracil compound. Thus, we predict that the 5-fluorouracil, just like the uracil, will attach itself to a sugar phosphate residue and then fit into the same place on the enzyme the uracil would have used. But once in place, the strong bond to fluorine will prevent the enzyme from replacing the fluorine with a methyl group. And finally, we predict that the strong polarity of the 5-fluorouracil will cause such strong adherence to the enzyme that the normal uracil compound will be prevented from reacting, and that, with the enzyme blocked, the essential chromosomal material cannot be made, and the cell will die. Let's test this prediction in the laboratory. Here are two cultures of cancer cells. Those on the left have been treated with 5-fluorouracil. Compare the action of these cells to that of the untreated cells on the right. The untreated cells continue to show rapid growth. The 5-fluorouracil treatment is seen to stop cell division, produce cellular enlargement, and eventually contraction and death. But are we sure that the cancer growth is stopped in the manner we predicted? Instead of the 5-fluorouracil blocking the enzyme as we have suggested, and using this means to kill the cell, is it possible that the 5-fluorouracil is incorporated into the DNA, inhibiting its function? To determine where the 5-fluorouracil is acting, in the enzyme or the DNA, let's conduct an experiment on some human cancer cells grown in the laboratory. A sterile pipette is used to transfer the cells into a large flask which contains a nutrient medium. This permits continued growth so that we will have enough cells for our experiment. The cells are kept in suspension by a magnetic stir and incubated for several days. Here is specially prepared 5-fluorouracil containing radioactive carbon-14. By tagging the 5-fluorouracil with these radioactive atoms, we can determine where the 5-fluorouracil goes in a human cell. We'll add a small amount of the radioactive 5-fluorouracil to the living culture. In about half an hour, these radioactive molecules will be incorporated into the cancer cells. The cancer cell suspension is poured into large cups to be spun in a centrifuge. The spinning collects the suspended cancer cells into a pellet on the bottom of the cup. The liquid is discarded. By this means, the cells from large volumes of the culture medium are finally concentrated into one cup. The cells are then resuspended in cold sodium chloride solution to dissolve the DNA. The low temperature retards enzyme action. This cell suspension is placed in a blender. The violent action of the blender fragments the cells to facilitate dissolving DNA. After storage overnight in a refrigerator, the DNA dissolves, and then the solution will be spun very rapidly in a centrifuge. This spinning action separates the residual cell proteins which contain the enzymes. A high-speed centrifuge is required because the large size of the DNA has made the solution viscous. After the centrifuging, the liquid fraction contains the soluble DNA. The pellet contains the undissolved residue. According to our theory, if the 5-fluorouracil is incorporated into the DNA molecule, radioactivity should be detected in the liquid. Otherwise, it should be found in the pellet. The next step is to concentrate the DNA. The liquid is drawn off and injected into alcohol, which precipitates the DNA. These threads of DNA material can be readily collected on a glass stirring rod. A sample of the DNA fraction is removed for measurement of its radioactive content. The protein pellet is retrieved from the centrifuge tube. The meter shows that there is no radioactivity in the chromosomal material. However, the protein fraction does show radioactivity. The experiment proves that the radioactive 5-fluorouracil is not incorporated into the DNA. Though the chemistry is oversimplified as portrayed here, it does show that the structural similarity of 5-fluorouracil to uracil is of crucial importance in its ultimate effect, the enzymatic synthesis of genetic material. 5-fluorouracil is currently one of the most effective chemotherapeutic agents for treating certain types of skin cancer and colon cancer. About 40 anti-cancer agents are in clinical use today. For example, chemotherapy with methotrexate can lead to complete remission for 97% of children treated for acute lymphocytic leukemia. But we need even more effective and less toxic chemicals, especially for lung cancers and brain tumors. In the past, most medicines have been discovered by trial and error, even by accident. Aspirin, the most widely used medicine in the world, was used for almost 100 years with no idea as to how it reacted in the body. Modern techniques are allowing us to learn about living systems at the molecular level and what changes cause loss of health. Now some mechanisms are understood. We are beginning to be able to design molecules the living system needs for recovery.
Okay. So that was that video. You can imagine writing a lab report for that, right? The details that you'd include in there. You know, it's the same kind of thing. The people are very curious about your sulfate analysis and want to understand the details of the analysis. You know, why you did certain steps. So think about that when you're writing up the discussion section. All right, we're going to check out now. And so go through your inventory. If you marked on your glassware with a Sharpie pen, I'd like you to remove the